What we're going to talk about today is topic 2.4, proteins. So what I want you to think about really fast is, what do you already know about proteins? Usually what comes to mind is muscle, is what a lot of times people think about, eating proteins to make muscle. But there's a lot more to it, and I know that we talked about it in 10th grade, and if you remember, we talked about how the cell's end product and goal is to make a protein. The proteins are made from DNA after protein synthesis, so transcription and translation. And we've also said what I always say over and over again, structure determines function of a protein. So let's get into this. So the first understanding is amino acids are linked together by condensation reaction to form polypeptides. So we've talked about a hydrolysis reaction. This is the opposite. We've also talked about a condensation reaction. But remember, what that is water being taken out, so two H's or two hydrogens and an oxygen being removed, and it forms a bond, which we call, because it's a protein, a peptide bond. So these would be two amino acids together, and you can tell they're amino acids. You see the R symbol right here, which is representing their unique amino acid state. And once this water is removed, these two amino acids bond together and it creates a polypeptide. So these are amino acids individually. Condensation reaction occurs, it creates a polypeptide, which is a chain of amino acids. Here is the monomer of a protein or a polypeptide, and you can see it always has the amino group, a nitrogen with two hydrogens, and then a carboxyl group on the outside of it. Then a hydrogen coming off the carbon, and then the R group, or the side chain is what it's called, and you need to know those for a test. And the side chain, you don't need to know them specifically, you just need to know what the R represents, okay? So what the amino group is, the side chain, and the carboxyl group are. You need to be able to label that. Okay, so there are 20 different amino acids that the human body uses, and we get it from our diets. We can't get it from anywhere else. And we build our proteins using these 20 amino acids. So here's a picture of the 20 amino acids. In the green part that's circled is their R, or their side chain. Okay, so notice they're all unique from each other, but the base of the amino acid in the amino group, the hydrogen, and the carboxyl group are all the same on each of them. Amino acids can be linked together in any sequence, which gives a large range of polypeptides. And this is insane how many combinations or different orders that the amino acids can go in to create a unique protein or polypeptide. So remember from the chart, I think from 10th grade, you guys used a codon chart. And that codon chart was things like gly, ala, val, met. Okay, those are just the acronyms or the short term for that amino acid. And the amino acids are put together based on whatever the DNA code told it to be, right? And then the tRNA brings that amino acid that matches that codon at the ribosome to do translation. So these are the 20 that it can be, and it can go in any combination. So it could be gly, ala, sur, or sur, lu, asp, or glu, lys, gly. So you can see there's so many different combinations. If you look here at this chart, it really puts it in perspective for you. So for every one amino acid, there's 20 potential combinations that there can be, right? Because it's only one amino acid. But if there's two amino acids, there's 400 different potential orders or combinations that can be created. If there were 3 amino acids, so you would go, think about it this way, 20 to the power of 2, 20 to the power of 3. You're going to get 8,000 possible combinations. If there's 4 amino acids, you're going to get 160,000. And if you look here, a typical protein is 300 amino acids. So if you put 20 to the 300 power, that is the number of atoms in our universe. So there are tons of possible combinations. So that means there's lots of different proteins that living organisms can make. And these 20 amino acids are not just in humans, they're in all living things. And so we have a lot in common with lots of different species because of that. So the amino acid sequence is coded for by genes, so remember it's actually DNA because genes are specific lengths of DNA. They're a code for like this piece of DNA, so it could be up to like, let's say 200 bases or longer, and that gene is called a gene, and that gene codes for a certain trait or protein. So let's say that the length of DNA or this gene is for eye color, and it's going to then lead to an amino acid chain that makes the proteins in your body to make your eye color, like blue or brown. So this is another chart that shows the most common 20 amino acids, and you can look at it. I thought it was just a good visual of it, but you can see the names of each of them, and these are the different codon combinations that would go with that amino acid. And you can see the codes. This is a little bit more about their environment that they usually are in, because that's important too when it comes to proteins, and we'll get into that more later. So I bolded this here, shape determines functions. And with proteins, this is the most important thing because proteins each have their own individual function, and that shape that they were coded for is what's going to be, or those amino acids, the combination of amino acids is going to create a certain type of shape, and then that shape can do its function. So a single polypeptide or more than one polypeptide linked together can be a protein. So this image here is 4 separate proteins, and so this is a very complex polypeptide or complex protein in its shape, so it probably has a more complex function, and we call this a quaternary structure when there are more than one protein attached to each other to make a more unique shape. So think about that. This one protein by itself, this red one, has a unique shape, which gave it a unique function. But when it's attached to other proteins, it can do even more functions. So it kind of blows my mind when we think about this, and it's all going on in our bodies, and we don't have to even think about it. So this next part is extremely important for you to understand how the shape occurs. So the amino acid sequence, so the gly, lu, met, all of those put together creates a chain, okay? And the order of those amino acids and their R groups coming off of them is going to create interactions between them. So this is a picture of a chain here. This would be an amino acid chain, this purple line. And each of these R groups coming off of the amino acids as they're linked together are going to interact with another amino acid, and they're going to be attracted to each other, and that's going to cause it to fold in a unique shape. So hydrophobic interactions usually happen between non-polar amino acids. So that would be these two R groups. They're non-polar, they're hydrophobic. So notice they fold inward because they don't want to be around the external environment, which is probably water or a liquid. So they're hydrophobic. There's also hydrogen bonds between polar amino acids, which are usually going to be a hydrophilic interaction, right? They want to be near water, so they usually fold and show themselves to the outside. So they kind of create these outside edges here on these proteins, are usually these hydrogen bond interactions. Ionic bonds are down here, and ionic bonds happen between a metal and a non-metal, and it's when they share, basically one of the elements or molecules takes the remaining extra atom or electron that's in their outer valence shell, and so they take it from it, and it creates this charge interaction between them. So they were unbalanced, and they're interacting between each other because they weren't balanced. So there was one extra one in one valence, and the other one needed an extra one to complete their valence, so it takes it. And that's that kind of interaction. Covalent bonds would be here, where we call this a disulfide bridge. And covalent bonds usually happen between the sulfur-containing amino acids. And they create this unique shape that we can't always predict, but we can learn so much from it once we figure it out. And we can figure out the proteins that are sticking off of viruses, and we can figure out how to break down viruses like the COVID virus, and that's what vaccines are trying to do. So understanding these interactions that are happening in the amino acid chain can lead to a lot of medical discoveries. Each individual, so each person, has a unique proteome, or each living thing. And in a proteome, so it kind of sounds like the word genome, like the Human Genome Project, which is all the DNA in a person. A proteome is the entire complement of proteins that can be expressed by a cell, let's say if the organism's only one cell, a tissue, or a full organism like us. We call that a proteome. So just always remember that shape determines function, and the amino acid interactions are what make that shape for a protein. So when a protein folds, it goes through 3 stages no matter what to be an active protein. So the primary, secondary, and tertiary stage is what's going to create a full protein that actually can do a function. If the protein then connects with another protein and makes a quaternary protein, then that'll still be very functional and complex. But you need to have at least the tertiary stage to be considered a functioning protein. So first, the amino acid chain is made through translation at the ribosome. Then it starts to interact, and it starts to fold either into a B-pleated sheet, like this red line, or an alpha helix, like this blue line, which is like DNA when it spirals. Then it starts to interact and have those hydrophobic, hydrophilic, covalent, and ionic interactions, and it creates a unique shape, and we call that a tertiary protein. And now that protein is functional, and it can do its job because its structure determines its function. So living organisms synthesize many different kinds of proteins with a wide range of functions. So just to name a few, structural proteins like collagen, this picture shows that. It's what makes our skin stay tight. Spider silk is another example of a structural protein. Contractile proteins like actin and myosin, which is in our muscle tissues and helps us move. It makes our muscles create action. Hormonal proteins like insulin, which is a hormone that helps decrease glucose in our blood, so our blood sugar levels decrease. Transport proteins like hemoglobin, which is your blood, which transports things like oxygen and nutrients throughout your blood system and gets them back to the organs that they need to be at. Protective proteins like antibodies or immunoglobulins, which are part of your immune system. Enzymatic proteins, there are so many of them, and they are what catalyze the reactions in your body, like amylase, which helps break down starch, and rubisco, which is in plants and helps them fix the carbon dioxide from the air and use it during photosynthesis. This is one of the most ancient enzymes of all time, rubisco. And then pigments. Rhodospins, which are photoreceptor cells in your eyes, and they help detect light. These are all different types of proteins in your body that fall into these categories. So there's tons of different other proteins in each of these categories. Some that IB really wants us to tell you about are on the next few slides. So rubisco is one. This is the structural shape of rubisco. This is filled in, so you can't see the interior connections, or interactions, but we always want to show you the structure of a protein because it's what allows for its function. So the full name of rubisco is rubulose biophosphate carboxylase, but you only need to call it rubisco. It's an enzyme that catalyzes or speeds up the reaction of fixing carbon dioxide in the atmosphere, and it provides the source of carbon from which all carbon compounds, which is required by living organisms, are produced. So without this, plants wouldn't be able to do photosynthesis. We wouldn't be able to then get the carbon that plants are made of as a food source, or oxygen as the waste product of plants as well that we breathe. It's found in high concentrations in leaves and algae cells. Insulin. Insulin is a hormone protein. It signals many cells, like the liver cells, to absorb glucose and help reduce the glucose concentration in the blood if there's too much. Affected cells have receptors sticking off of them, like proteins, that are proteins as well, on their surface, which insulin can then bind to it. So the shape of insulin, which you can see here, binds to those receptors on the cells, and then it triggers those B cells in the pancreas, and it transports it back through the blood. So it signals it to secrete insulin by the B cells, and then from the pancreas, and then the insulin will be transported through the blood and pick up the glucose. So the pancreas of a type 1 diabetic doesn't produce sufficient insulin. So this would be type 1 diabetes. And therefore, sometimes you might have to take, that's what this picture is of, synthetic insulin that's man-made to let the body start picking up that glucose from your bloodstream. But the important thing to realize is that the insulin, the shape of it, is what's going to trigger the body to release that. Immunoglobulins. So immunoglobulins are part of our immune system. So they're a protein known as an antibody. Antibodies always have a specific shape on their arms. Out here, this is an antibody, which is going to match with different pathogens or antigens that try to interact in our body. This is the protein shape in 3D. This is a 2D image of it. But there is something that these ends bind to specific antigens, which let them be aware of different pathogens or diseases that might be bacteria, viruses, that might be trying to enter your body. And they've memorized it from past interactions, and then they can trigger other things like immunoglobulins to then they trigger immunoglobulins, which are then going to act as a marker to a phagocyte, which is a large organelle in your cell that's going to engulf that pathogen. It's going to eat that pathogen before it hurts your body. So antibodies and immunoglobulins are examples of your immune system's proteins. Rhodospins are what I had talked about before. It's a pigment that absorbs light in your eye. So this is its structure right here, and they kind of look like long rods in the shape when you put them all together, so that's part of their name. So this membrane protein of rod cells of the retina in your eye, and so it's a light-sensitive region at the back of your eye where these proteins are located, and even very low light intensities can be detected because of this protein. So it's what helps us see. Collagen. So there's lots of different types of collagen throughout our bodies. It's not just in our skin, but you can see here the shape. It's kind of this meshy, long, circular shape, and it's rope-like proteins, which are made of 3 polypeptides. So here, 3 amino acids. 3 proteins. So you can see 3 different colors here. So 3 different proteins. This would be a quaternary structure protein. And about a quarter of all proteins in the human body are collagen. It forms a mesh of fibers in your skin and blood vessel walls that will resist tearing, so it helps you be more flexible. It gives strength to your tendons, ligaments, skin, and blood vessel walls, and it forms part of the teeth and bones, and it helps to prevent cracks and fractures to bones and teeth. Spider silk. Spider silk is a unique structural protein as well, like collagen. What's unique about it is it kind of looks like a mess here. This is one unit of it, but when you look at it more closely, when it's stuck together, it looks very disordered and tangled, but when it stretches out, it becomes more straight, so you can see how they're all linked together here, this loose area. But when it stretches out finally, it makes it actually more strong. It's like a mesh net, and it makes it more extensible and very resistant to breaking. It's similar to Kevlar, which is like when you think of like seatbelt material. So the last part of this is proteins do have a problem. They can be denatured. So denaturing of proteins is when something in the environment makes the protein lose its shape. So it unfolds. And a lot of times it's permanent, right? So a denatured protein does not normally return to its former shape. Soluble proteins often become insoluble and form a precipitate. So you lose the protein for good when it becomes denatured. So all proteins want to be in an environment that they can be successful at. So they want the pH to be a certain pH, like acidic or basic or neutral. And if you go out of those pH ranges, it'll then denature that protein. So it's going to change those R groups, and it'll break those ionic bonds that are in there, because it might change their charge, and causing different ionic bonds to form. So now the shape changes, or it unfolds all the way, and now it can no longer do its original function. Temperature can also do this. Usually it's heat. So proteins like to be in a certain type of environment with a specific heat, and when they get out of that range, they then unfold. Those bonds break because of the heat, and they unfold, and then they no longer have that structure, so they lose their function. So those are the two ways that a protein can become denatured. So when someone's really sick and has an extremely high temperature, you can imagine what all the proteins in their body are starting to do when it becomes like a deadly high temperature. So that's one thing there. And think about all the foods and different toxins we might eat that have extreme pHs. That's a reason why that can hurt our proteins as well. So that's it. We're going to stop there for the day. Please put your notes in ManageBack and have your questions ready because we can address them at the beginning of next class. Let me know if you have any questions, and I hope this was helpful.